Hello everyone, I'm from MyTech. I'm very glad to share with you the topic of how to solve the problems of health of liver and intestine of aqua in this way. Today, I will introduce in four aspects. First part, we will discuss the role of health of aqua liver and intestine in improving the benefits of aquaculture. The second part, I will introduce the performance of Fortide in improving the health of liver and intestine, and then introduce the application of Fortide in aquafeed. Finally, I will give you a brief introduction of the production process and its corresponding indicators and evaluation methods of Fortide. So first, let's discuss that why we say promoting the health of liver and intestine is a key measure to improve the benefits of aquaculture. As we all know that farmers As we know that farmers they concern about the yield and the benefit. That means they care about how many aqua and how much money they can get from the pond, and also they care about the poor resistance and transport resistance of the aqua while selling and transportation. So let's analyze these three factors one by one. The first one, the yield per hectare. This is based on the seeding quantity, the weight within specified time, and the survival rate. The seeding quantity is determined by the farmer based on the actual load of the pond, which is basically a relatively fixed value, and the body weight in specified time is affected by the growth rate, that is, the feed utilization efficiency, and the survival rate is determined by the ability of anti-stress, that means the physical health of the aqua. So we can say the feed utilization efficiency and the physical health of the aqua is important factors for production yield. The second is the benefits. This is dependent on the yield, fish price, and feed consumption, medicine cost. Yield we just mentioned just now, and for the feed consumption, this is mainly determined by the FCR. The cost of medicine for water quality adjusting is affected by the water quality. We know that reducing the emission of the nitrogen and phosphorus will help improve the water quality and save the cost of the medicine. The third is poor resistance and transport resistance. This is related to the anti-stress ability of aqua and the anti-stress ability is closely related to the health of functional organs, especially liver and intestine. By the analysis of the economy and concern of farmers, we sum up the key points are the digestibility of the raw material, digestion ability of the fish, and this is related to the development and health of the intestine. And the second is the healthy liver. We know that liver is the main organ for metabolism and the detoxification organs. And therefore, in order to meet the needs of the aqua farmers by feed technology, the key is to focus on the following two aspects. First is to improve the FCR, and the second is to keep the health of the functional organs like the liver and the intestine. And in order to improve the economy benefits of the aquaculture, it stimulates the speed up of the improvement of the breeds. So the growth potential of aqua has been greatly improved, but their digestion and absorption capacity and immunity have declined. The main reason is that the development of functional organs cannot keep up with the speed of growth. And these problems reflected in productions are poor disease resistance, high morbidity, high mortality, poor anti-stress ability, low poor resistance, transport resistant. In addition, the nutritional level of aqua is generally high, which increases the digestive burden of aqua and increases the emission of the nitrogen and phosphorus, and also worsen the water environment. The deterioration of the water environment further affects the health of the functional organ of aqua. And there is a saying in China in aquaculture, we say that good farming of aqua is to farm the intestine, liver, and pancreas of aqua. So this reflects the importance of health of liver, pancreas, and intestine to the overall physical health of aqua. So you must wonder what are the most direct viewing of liver problems in production. One is that the color of liver changed. For example, the normal color of liver is blood red. For the first picture, this is the liver of grass cap, and the color changed into white. And the second picture, this is a liver of catfish. The texture of liver becomes spotted, and some white spots appeared. And this is the liver of California bass. Its color changed into yellow. And the second viewing is abnormal size of a liver. The liver is too large, too small, or atrophied. The third is lesion, for example, yellow nodules and bleeding spots on the liver. We know that the health of liver is very important, and its meaning depends on the function of liver. So first, the liver participates in the metabolism of sugar, protein, fat, vitamins, hormones. It has important function of metabolism. The second is that the liver breaks down sugar, stores glycogen, has the function of storing nutrients. And the third is that the liver is the largest reticular endothelial cell phagocytosis systems, and 99% of the bacteria in the bovine blood are swallowed when passing through the hepatic vein sinus, which has a strong function of immunity. The fourth, the liver is an important organ secretes bile juice. The secreted bile juice can promote the digestion and absorption of fat in intestine. In addition, the liver also has the function of synthesizing coagulation factors. When the liver is diseased, which will lead to a lack of coagulation factors and cause an increase in clotting time and the tendency to bleeding. This shows that the health of liver is very important and plays a central role in the healthy growth of aqua. So, how to solve the liver problems during the farming process? First, we need to analyze the main mechanism of liver problems. There are many influence factors that cause liver problems. Here, we divided them into nutritional factors and non-nutritional factors. The problems caused by nutritional factors are fatty liver problems. Let's see the mechanism on the left. We know that high nutritional level of protein and lipid and overfeeding often result in the high energy intake for aqua, and long-term high energy intake can lead to an increase in plasma free fat acid in aqua, and this free fat acid enters the liver through blood circulation. There are two main metabolic pathways of the entered fat acid. First is the long-chain fat acid was transported to the mitochondria by calcium transporter for energy supply for beta oxidation decomposition, and the second is synthesized triglycerides and formed fat droplet in liver cells. The triglycerides can only be transported out of the liver by passing very low density lipoprotein we called VLDL, and if these two pathways are blocked, On one hand, excessive accumulation of fat acid will produce the lipid toxicity, which cause the rise of the inflammatory factors and cause damage to liver cells. On the other hand, synthetic triglycerides cannot be transported out of liver and leading to fat liver. Meanwhile, too much triglycerides will introduce oxidation stress, cause damage to liver cells, and even lead to liver fibrosis and liver necrosis. And as for non-nutritional factors, there are the oxidation factors in fat and the environmental stress factors and also the toxin in the fat. For the oxidation factors in fat, such as the oxidation of a protein and lipid, for the environmental stress factors such as water temperature, ammonia, nitrogen salts, and medicines. And all these factors will lead to damage to the structure of liver and cause the liver dysfunction and affect the oxidation decomposition of the fat acids and the transfer of triglycerides, and even lead to the fat liver, liver fibrosis, and liver necrosis. Through the analysis of the main mechanism of liver problems, we can get that to solve the problems of liver, we should focus on the following aspects. The first is to design a balanced and effective nutritional level. So the level of protein, fat, sugar should be balanced. And also it is important to select the right source of raw material to avoid the excessive energy intake. It is worth thinking that how to improve the digestion and utilization of nutrients under the nutritional level. I think there are two aspects involved. One is to consider the digestibility of the raw material, and the other is to improve the conversion efficiency of the nutrients. And this is closely related to the development and health of the intestine. The second is to strictly control the quality of the raw material and reduce the introduction of toxical and harmful substances into the feed, such as reducing the oxidation of animal protein and lipid to minimize the bad factor like the various anti-nutritional factors brought by the protein source, and strictly control the mycotoxins of raw materials. The third is to improve the oxidation of fat acid and the transfer of triglycerides, reducing the risk of fatty liver. We know that without the transfer of collagen transporter, fat acid cannot enter mitochondria for beta oxidation, and the L-carnitin is the core substance for collagen transporter. The raw materials for the endogenous synthesis of L-carnitin are lysine, the carbon source, and the methionine, the methyl source. L-carnitin is synthesized under the action of lysine, folate, vitamin B6, vitamin C, and some related enzymes. Therefore, sufficient L-carnitin, lysine, methionine should be provided in feed to ensure the normal synthesis of L-carnitin. For ensuring the normal function of the collagen transporter, the transfer of triglycerides requires the transfer of very low-density lipoprotein. The VLDL is mainly composed of ApoB and phospholipids. Many nutritional factors can affect the synthesis of lipoproteins. Therefore, it is necessary to ensure the sufficient content of the key substances in the synthesis path, such as phospholipid, choline, and methyl salts, and so on. The fourth is to enhance antioxidant capacity of the liver and alleviate the damage caused by the bad factor to liver cells. The common antioxidant substances in fat, such as vitamin C, vitamin E. The fifth is to reduce the environmental stress factor through the refined management, such as a reasonable stocking density, pay more attention to the water quality, reducing the use of the medicine, especially the antibiotics and the sulfa drugs. After talking about the liver, let's look at the intestine. The direct effect of the intestinal problems in production are bleeding, thin walls, poor toughness of intestine, and the intestine is very easy to suffer from enteritis and intestinal sepsis and interceptions and other diseases. We know that the intestine is the main organ for digestion and absorption of the nutrients, and it is also the first line of defense for aqua against the invasion of toxical and harmful substances from the outside. And the intestinal injury usually causes damage to liver and other organs of aqua, which is the initial organ that causes damage and dysfunction of other organs. So the normal growth and development of intestine and integrity of its morphological structure are the basis for all function of the intestine. From this, we can see that the health of aqua's intestine is very important. And compared with land animal, aqua intestine have their own characteristics. First, the structure of the wall of intestine is thin and vulnerable to damage. The following two figures visually show that the differences between the structure of aqua intestines and piglet's intestine. There is no obverse submucosa on the structure of the wall of aqua intestine, and the intestine canal of land animal consists of mucosa layer, a submucosal layer, a muscular layer, and a serosal layer from the inside to the outside. The tissue structure of the intestine tract of the bunny aquas is very similar, generally including the mucosal layer, muscle layer, and the serosal layer, and the thickness of each layer is thinner than that of land animals. Therefore, the structure of the intestine wall of aqua is thin, and only 20th of the thickness of intestinal wall of land animal. For example, the thickness of the jejunum wall of piglets is 2,271 microns, while the thickness of the intestine wall of tilapia is only 92 microns. Therefore, we can see that aqua intestines are extremely vulnerable to damage and destruction. The second is that the high content of polyunsaturated fatty acid in the aqua intestine, which is vulnerable to oxidation damage. The content of the polyunsaturated fatty acid in the intestine of aqua is much higher than that of land animals, such as piglets and carps. The content of the polyunsaturated fat acid in the intestine of pig is 34%, and that of carp is 49%. Therefore, aqua are susceptible to oxidation damage of peroxide in the feed, such as oxidative rancidity of the fish meal and oxidation of the lipid. Generally speaking of the intestinal health, everyone will talk about the health of the four major barriers, including microbarriers, chemical barriers, physical barriers, and immune barriers. For the microbarrier is composed of pathogens, conditional pathogens, and symbiotic bacteria. Under the normal condition, the types and numbers of these three types of bacteria are in a balanced state, but due to the poor digestion absorption of the nutrients that are not digested and absorbed will be fermented by the microbes and change the environment of the intestine, including the osmotic pressure, pH value, and oxygen content. So the environment of the intestine will become more beneficial to the development of the pathogens and lead to a mass production of the pathogen and then attack the intestinal epitheliums and break the balance of the microbes in the intestine. And the poor digestion absorption are caused by two factors. One is the digestibility of the raw materials, and the other is the digestion and absorption capacity of the animal themselves. And for the digestion and absorption ability of animal is mainly based on the physical barriers because this is a place where digestive enzymes are secreted and the nutrients are absorbed. So to a large extent, the microbial barrier is affected by the physical barriers. And the chemical barrier is between, or we can say in the middle of the microbial barriers and the physical barriers, and its components are from the products of these two barriers. It cannot exist independently. It is affected by the two barriers. So we can conclude as among these four barriers, the physical barriers and immune barriers are more important, and therefore, either judging from the status of the intestine of aqua or importance of physical and immune barriers in the four major barriers of intestine, it is necessary to strengthen the function of these two barriers. So how to strengthen the function of these two barriers? For building a better physical barriers, the cells need to quick proliferate and differentiate because the cells here are renewed very quickly, and the second is that the structure of cells must be completed to maintain their normal barrier function, and the third is that the tight conjunction between cells and cells must be high, that is, permeability should be low to keep the bacteria, toxins, harmful substances outside the intestinal epithelium cells and enhancing the function of the physical barriers. If some toxic and harmful substances enter the intestinal tracts through the physical barrier, and the macrophages will swallow them. This is the swallow function of the macrophage, and in the progress, there are two quick reactions. One is the inflammatory response, and the other is the production of a large number of free radicals. Therefore, to enhance the immune barrier function of the intestine, we must pay attention to the function of the anti-inflammation and antioxidant. From the above analysis of the influence factors on the health of liver and intestine, we can have the following conclusion that in order to improve the competitiveness of aqua feed, we need to improve the disease resistant and anti-stress of the aqua while we ensure the good production performance, and we can work on the following aspects to design a balanced and effective nutritional level, control the quality of the raw material, adopt the innovative and healthy feed technology, and the refined management. For the innovative healthy system technology, we must pay special attention to the promoting the health of the liver and intestine. For the liver, we can think about how to promote the oxidation of the fat acid and the transfer of the triglycerides, and how to enhance the antioxidant capacity of the liver. For the intestine, we may think about how to enhance the function of the physical barrier and immune barrier of the intestine. So, based on the study of the point of improving the health of aqua liver and intestine, MyTech use soybean meal and soy protein concentrate as raw materials, designed our own production process of enzymatic hydrolysis, and get our product Fortide. On one hand, Fortide is rich in small molecular active peptides, which is less than 1,000 Dalton, and this is a small peptide of 226 amino acid, and this small molecular active peptide have the function of promoting the quick proliferation and differentiation of the intestinal epithelium and increasing the tight conjunction between cells. Also have the function of antioxidation and reducing the inflammatory response, therefore enhancing the function of the intestinal physical barrier and immune barrier. And these small molecular active peptides, many are a chain of primary structure, so the high temperature, high humidity, and extrusion will not change their structure. So the processing of the aqua feed will not influence its function. On the other hand, in the process of enzymatic hydrolysis, the large molecular becoming small moleculars, it effectively eliminates the anti-nutritional factors, and the elimination rate is more than 95%. Therefore, Fortide also has the feature of high digestive utilization and with low anti-nutritional factors. We know that anti-nutritional factors will cause the bad effect on the physical barrier of intestine. So, what's the performance of Fortide in improving the health of liver and intestine in aqua? Let's look together. First, Fortide can significantly enhance the antioxidant capacity of the liver. The research was done on caps with Sichuan Agricultural University. The test is to select the cups with an initial weight of about 12 grams as the test object. Reduce the protein level by 2% on basis of the original doubt. So the 34% CP doubt as a positive control and 32% CP doubt as a lactive control, and we add different level of Fortide on basis of the lactive control: 1%, 1.5%, 2%, and 2.5% and 3%. And this trial lasted for 56 days. From this result of the study, we can see that in the 32% CP plus 1.5% of Fortide group, the content of oxidation products of the lipid, MDA, and the content of the oxidation products of the protein carbonyl had decreased significantly. Compared with the 34% CP positive control group, they decreased by 17% and 9% respectively. And also the activity of antioxidant enzymes in the liver, such as the CAT, SOD, GST, are also significantly increased. The above results show that Fortide can significantly improve the antioxidant capacity of the liver and alleviate the influence of the bad factors on the liver. The application of Fortide can promote the health of liver in actual production. Here is an example of Fortide application result in catfish feed from a Sichuan company. We use Fortide in this catfish feed less than a month, and the proportion of the spotted liver and white liver was significantly reduced. And at the same time, we observed that the proportion of visceral fat of the channel catfish decreased significantly by 12%. And this shows that the health of liver can promote the fat metabolism. Also, the reduction of the visceral fat can improve the aqua's resistance to pull and transportation. And we also got a similar result in cat feed from a company in Liaoning. The ratio of fat to viscera was 18% in the control group, and that was 11% in the Fortide group. There is a decrease of 7%. And from the above result, we can see that the application of Fortide can promote the health of liver, reduce the deposition of the visceral fat in aqua. So after talking about the liver, let's see the intestine. What are the effects of Fortide on enhancing the health of intestine of aquas? Firstly, Fortide can enhance the physical barrier function of the intestines. This below study on caps found that Fortide can promote the growth and development of intestine. And we saw that the weight of intestine, the intestinal index, and the height of the folds of the foreguard, middle guard, the hind guard all increased significantly. Ensure the better development of the intestine helps to improve the digestibility of the feed. And in the actual aqua farming, we also found that the growth of the intestine is better after using Fortide, and we usually use the weight of intestine to measure the health and development degree of the intestine. The heavier the intestine is, and the better the development of the intestine. So after adding Fortide in catfish feed, the weight of intestine per unit length has increased by about 20%, and there is low bleeding point in the intestine, and the intestines are rich in folds. This helps digestion and absorption of the nutrients. And the result of promoting the development of intestine is also consistent with the result we got from a company in Meishan. After 3.5 hours of feeding, the feed was fully digested, and the chyme was mainly concentrated in the posterior of intestine. We dissected the intestine and observed that the intestine was healthy, without bleeding point and rich in folds. And the second is that Fortide can improve the digestion and absorption capacity of the intestine, thereby improving the feed utilization. From this study on caps, we found that compared with the control group, the activity of protease and lipase in intestine in the 1.5% Fortide group was significantly improved, with the range of increase by 7 to 20%. And the function of Fortide to increase the activity of digestive enzyme has also been confirmed in study on groupers. This research was conducted at Guangdong Ocean University. The groupers with an initial weight of about 12.5 grams was selected as test objects, and the original basic diet was with 48% CP as a control group, and on the basis of the control group, we add 4 different levels of Fortide: 1%, 2%, 4%, and 6% of Fortide. And according the different level of Fortide, reduce the half imported fish meal and half dehued soybean meal. After 56 days, this test results show that for the addition of 4% Fortide group, the activities of trypsin, amylase, and lipase increased significantly. Trypsin's activity increased by 48% and lipase activity increased by 83%, and lipase activity increased by 123%, and the improvement is very significant. And based on the better growth and development of the intestine and the increased activity of digestive enzyme, the ability of digestion and absorption of nutrients is enhanced. The digestibility experiment we done in our laboratory also proved that the apparent digestibility of the protein can be increased by 4%. And in actual farming, we determined the protein content in feces, and it showed that the application of Fortide can reduce the protein content in feces by 1% and improve the utilization of feed, as well as reduce the emission of the nitrogen. And third, Fortide can increase the density of the intestinal epithelium by measuring the expression of the tight conjunction protein of foreintestine and middle intestine of calves. We see that the genes expression of tight conjunction protein increased by 20 to 40%, therefore enhancing the density of the intestinal epithelial cells and reduce the permeability of the intestinal epitheliums and effectively prevent the invasion of the pathogenic microbes. And in addition, Fortide enhances the function of the immune barriers. On one hand, after using Fortide, the relative expression of intestinal pro-inflammatory factors genes IL-1 beta TNF-alpha was significantly reduced, and while the expression of the anti-inflammatory factor genes TGF beta 2 and IL-10 increased significantly by 10 to 25%. This showed that after applying Fortide, the ability of anti-inflammation of the intestine is significantly enhanced. On the other hand, because Fortide is rich in antioxidant active peptides, it can significantly enhance antioxidant capacity of the intestine, and this is proved in this experiment by the decreased content of the oxidation products of intestine, MDA, and protein carbonyl, and the increase in the activity of the antioxidant enzyme. And we got the similar result in the study of grouper. After using Fortide, it can significantly reduce the content of MDA, increase the activity of antioxidant enzymes, and improve the antioxidant capacity of intestine. So, a summary of the previous content: Fortide is rich in active peptides that promote development and structural stability of functional organ, and is rich in antioxidant active peptides in vivo for intestine by promoting the proliferation and differentiation of the intestinal cells, maintaining the integrity of the structure of cells, and increasing the density of the intestinal epithelium cells, reducing the permeability. Fortide enhanced the function of the intestinal physical barriers, and by enhancing the ability of the anti-inflammation and anti-stress, Fortide enhance the function of the immune barrier of intestine. And for liver, by improving the antioxidant capacity of the liver, promoting the health of liver, protecting the function of liver, reducing the risk of fatty liver, and reducing the visceral fat deposition. This reflected in the production of farming is, on one hand, they are high feed utilization, fast growth, low FCR, and good water quality. On the other hand, for aqua, they are with healthier functional organs and higher disease resistant, lower mobility, and enhancing the ability of anti-stress, make aqua get a healthier resistance to pull and transport. In the third part, I will give you a brief introduction of the research and application of Fortide in aqua feed. As for common aqua farming, we have carried out research on caps and grease caps with Animal Nutrition Institution of Sichuan Agriculture University, and also carried out research on catfish with Aqua College of University of Agriculture and Forest in Vietnam. All the research results show that using Fortide reduced the cost of feed formula, and not only the growth rate was increased by 5%, but the FCR was reduced by about 5%, but got a healthier functional organ of aqua, and all the corresponding articles about the study was published. So in terms of the special aqua, we have carried out research on groupers and shrimps with Aqua College of Guangdong Ocean University. The research on groupers Fortide has significantly increased both physical and chemical indicators, as well as production performance. The activity of intestinal digestive enzyme is increased by 50 to 120%, and the intestinal antioxidant enzyme are increased by about 20%. The growth rate is increased by 5%, and the FCR is reduced by 10%. For shrimp, they conducted Vibro Harvey challenge tests to count the mortality rate in 7 days, and they got the conclusion use Fortide can improve the survival rate by more than 10%. And also we carried out an evaluation on the catfish in the net cage experiment based of a group company under the permission of the reducing the cost by 30 RMB per ton. The growth rate increased by 4%, and the FCR decreased by 3%. The above study and applications have confirmed that Fortide has a role of promoting growth, reducing FCR in aquaculture, and these are related to the effect of Fortide to promote the health of functional organs, especially the health of intestine and livers. And based on the research and application of Fortide, we give recommendation of the dosage and application. The recommended dosage for common aqua is 1 to 2%, and the dosage for special aqua is 2 to 6%. And there is two recommended application. One is to reduce the use of high-quality protein raw material or the similar functional products, and the other is to reduce the level of protein and fat in feed. For the dosage and usage of Fortide for specified species, our technical department will formulate the corresponding solution according to the needs of the customers. The second case was a study on catfish conducted by the Aqua College of University of Agriculture and Forestry in Vietnam. The animal stage is 14 to 130 gram juvenile catfish, and animal designs two treatment groups, 4 repetitions per treatment and 130 towels per repetition. The application is that we use commercial catfish feed with 30% of CP and 8% of EE as a control group, and on the basis of the commercial catfish feed, add 0.8% of Fortide and reduce the same level of fish meal. The inspection indicators are SGR, FCR, and survival rate. And this is the personnel involved in the experiments. And the experiment cage, the weight data recorded. You can take a quick look at the composition of the feed in this experiment. The research results were that Fortide group with the same growth rate and FCR reduced by 3% and also reduced the cost of feed by 0.1 RMB per kilogram. The third case is a study on a channel catfish in an experiment phase of a group company in China. In this case, the animal stage is 100 to 340 gram juvenile channel catfish, and animal design in 3 treatment groups, 3 cases per treatment, and 90 taels per cage. The application is that use commercial channel catfish as a control group, and on the basis of the commercial feed, reduce 2% of CP, add 1% or 1.5% of Fortide as the experiment group, and the inspection indicators are the weight gain, feed intake, and FCR. And this is the case experiment base of the group complete in Sichuan. This is a formula composition of each treatment group. The cost of adding 1% of Fortide group is 30 RMB per ton. The price is lower than that of the control group, and the cost of adding 1.5% of Fortide group is equal to that of the control group. We can see the research result for 1% Fortide group is that with a lower cost of feed and having the equivalent gross performance as the control group. For 1.5 Fortide group, getting an increase in the weight gain by 4% and a decrease in the FCR by 3%. And it is based on the remarkable effect of Fortide in healthy aqua farming. Our company has won many awards, among them the joint research project led by Sichuan Agricultural University. We joined the project linked systematic nutritional technology and application of healthy carp farming, won the National Science and Technology Progress Award in 2013 and 2019. This is an affirmation of the effect of our products, as well as an affirmation of our colleagues in MyTech for 17 years of consistent focusing on R&D and production of Fortide. So Fortide is an excellent product, and you must wonder how we made this product and how to evaluate its corresponding indicator and features. So let's continue to the following part. First, we use the whole soybean meal and soy protein concentrate as raw material to produce Fortide through the process of enzymatic hydrolysis. There are 2 important steps in the progress of enzymatic hydrolysis. The first step is to break the wall of cells so that the protein in the cells are fully exposed, which can fully interact with protease. The second step is the enzymaticase of the large molecular protein or peptides by different proteases, so to obtain more small molecular active peptides below 1,000 Dalton. And you may think this is a simple process, but before a large-scale production, our technical R&D department will do a lot of important basic research and preparation. The most critical is reflected in 2 aspects. The first, we need to fully understand the protein structure of the raw materials, and from the information in the figures, we can see that we have now mastered the primary structure of the soy protein, that is the amino acid sequence. And this provides an important theoretical basis for finding position where the small molecular active peptide exists and selecting the corresponding protein enzymes. The second important basic R&D work is to study the active characteristics of the protease. We know that the site of action of protease is specified, so after we confirm the sites need to be digested, we need to select the corresponding enzymes to achieve the process, and we own the intellectual property right of one enzymes. It is for obtain more effective small molecular active peptides, and also enzymes have important catalytic characteristics. That is to say, there is a lot of research work need to be done in advance, like the right number of substrates and enzymes to maximize the efficiency of the enzymatic hydrolysis, and also the enzyme has sensitivity to the conditions such as the temperature, pH value, reaction time, so we solve all the issues that ensure both the high efficiency of the enzymatic hydrolysis and also the stability of each batch of our product. After the basic R&D preparation work is done, the production process is actually relatively easier, which includes three parts. The first is to confirm and select the raw materials and other ingredients. We choose plant-sourced raw material because of its wide sources and high safety. The raw material and other ingredients enter the system of liquid enzymatic hydrolysis. The liquid enzymatic systems ensure the accurate amount of raw materials and other ingredients and the materials reaction in high quality liquid condition with monitoring of the reaction temperature, pH value, and reaction time. The fully enclosed reaction tanks without any contamination by the bacteria. The time of enzymatic hydrolysis reaction is 12 hours, and after this, it enters the post-processing systems like the drying, the crushing, and packaging. For drying, we adopt a low temperature drying to ensure that the quality of the intermediate products from enzymatic hydrolysis will not be damaged. And there are some pictures of our production equipments. You are welcome to visit our production sites. For the selection of raw materials and design of the process, we strive to achieve the safety and stability of our product. So we adopt ISO quality system and pass GMP Plus certification, and including the test reports from the third part, which demonstrates the biosafety of our product and its effectiveness. As we mentioned many cases of the application of Fortide and aqua, Fortide promotes the health of liver and intestine, increase the growth rate, and reduce the FCR. They are closely related to the characteristics of Fortide. We have also developed a set of detection methods for the main features of functional enzymatic proteins that can be quickly detected in the laboratory. The first very important feature is that the content of peptides below 1000 Dalton is very high, and this part of the peptide is an important component to play a functional role, and there are three different detection methods. The first method is the TCA precipitation method, which can roughly determine the content of peptides. From the data in the figure, it can be seen that the peptide content accounts for more than 28% of the entire substances and more than 62% of the CP content, which significantly higher than that of the other products like animal proteins and other enzymatic or fermented plant protein. The second method is SDS page electrophoresis, which is a method for qualitatively determining the molecular weight of protein in different substances. From this figure, we can see the protein in our functional enzymatic proteins are mainly concentrated in the region of 10,000 Dalton, while other animal proteins, enzymatic or fermented plant protein are in a large molecular weight range, but this does not mean that the molecular weight of our product is around 10,000 Dalton, because the protein raw materials of different molecular weight should be presented in the same page. And it is true that the protein molecular weight of our product is much smaller than that of other products. The third method is HPLC method to quantitatively detect the range of and duration of the molecular weight of protein. From the data in the figure, we can see that the range of Fortide below 1000 Dalton accounts for more than 54% of the entire protein and more than 24% of the entire substances. For the raw material, the dehued soybean meal, the range below 1,000 Dalton accounts for only 2%, and the range of fermented soybean meal below 1,000 Dalton accounts for only 2% too, but the portion below 10,000 Dalton has increased compared with dehued soybean meal. It shows that the fermentation process is helpful to reduce the molecular weight of protein to some extent, but not as good as our process of enzymatic hydrolysis. The second feature is that Fortide is with strong antioxidant activity. In the previous introduction, Fortide can enhance the activity of antioxidant enzymes in body to increase the antioxidant capacity of cells, thereby reducing the content of the products of antioxidant damage, MDA and protein carbonyl. Then in vitro, we can test its antioxidant capacity by ABTS method. For a better comparison of the ability of scavenging free radicals, we introduced the two well-known in vivo antioxidants, the vitamin C and vitamin E. From the data, you can see that the antioxidant of 1 kilogram Fortide is equal to that of 26 gram VE and that of 10 gram VC. And also we added these substances, Fortide, VC, VE, for production of aqua feed, once again determine the ability of this feed to scavenge free radicals. From the data, we can see that after adding the same amount of antioxidant VC and Fortide, the ability of scavenging free radical are significantly improved, and the antioxidant of Fortide group is significantly higher than that of VC group. It indicates that the production process of aqua feed will not affect the functional components of Fortide. The third very important feature is that the content of anti-nutritional factor is very low. The common anti-nutritional factors of soy protein are soybean antigens, including glycine, beta-conglycine, and trypsin inhibitors. We determine their content in different raw materials by ELISA method. From the data in the table, it can be seen that the emulation of the antigen protein of Fortide is over 95%, and which is much higher than other plant proteins. This can indicate that our product Fortide effectively handle ANF in raw materials without causing the bad effects on the intestinal physical barriers. And that's all for my introduction. Thank you for listening.